Welcome to the PRCCEH seminar. Today we have Dr. Joseph Romano. He is Assistant Professor of Informatics and Pharmacology at the University of Pennsylvania in the Department of Biostatistics, Epidemiology, and Informatics. So his research aims to discover and explain new associations between environmental exposures, including both social determinants of health and xenobiotic chemicals and human disease via secondary analysis of real-world human health data and structured biomedical knowledge. At Penn, he is the Associate Director of the Environmental Health Informatics Core in the SEED, where he maintains inventory of EHS-related data sources and software analysis pipelines. He also serves as Associate Director of Biomedical Informatics Education in Penn's Permanent School of Medicine. So this is great, Joe, and the floor is yours.
Great. Thank you so much. I appreciate the introduction, and I appreciate the invitation to come here and speak with you all today. So as you notice from the title, environmental toxicology is really one of the major spaces where I do my research, but one of the sort of bodies of indications we have been migrating into a lot, particularly with some of the students in my research group, has been maternal health and sort of perinatal health. So I'll be talking first about sort of more broadly applications in environmental toxicology and environmental health sciences, and then moving into some specifics in works that we are currently doing, have recently done, and plan to be doing in the near future, focusing particularly on adverse outcomes in maternal health. So let me go ahead and give you a spoiler. So the informatician in me is here to teach you that what we want to do, the mission of my research, is that we need to leverage knowledge and representations of knowledge, biomedical knowledge, to expand what we can do with data. All of us work with data on a daily basis in our research. And knowledge will help us to expand the capabilities of what we can do with it and also to increase our power. I don't need to spend a whole lot of time on this because most of it's already said, but this is an outdated picture of my lab that you see on here. We need to have another social event where we can show all the new members. Basically, our research sits at this intersection kind of between translational bioinformatics, biomedical artificial intelligence research, and environmental toxicology and other aspects of environmental health sciences as well. And I am involved in teaching. I teach Intro to AI, Intro to Python, which is, I've been doing that since I was a postdoc here at Penn. And I do guest lectures and other courses, both in the seat and outside of that. So the objectives for today's talk, I just want to run through them really quickly. First, I'm going to introduce kind of the role that artificial intelligence has in environmental health science overall. Then I'm going to get into this informatics-y concept of knowledge representation and how we can use this to do things better in predictive toxicology. Then I'm going to introduce this platform that we've been increasingly iteratively developing called ComptoxAI, and talk about its applications to predictive toxicology. Then I'm going to explore some of those specific projects we've been working on, particularly focusing on maternal environmental health. And then I will close out by getting on my soapbox for a minute or two and talking about what I think the future holds for AI, particularly in environmental health science, to give us some sort of guidelines about the future and how we might want to think about it. So I probably don't need to define this, but I like to do it just so we're all on the same page. I view environmental toxicology as the study of the adverse effects of chemicals, but also social determinants of health and other types of environmental exposures, which aren't listed here, on living organisms and the environment that those organisms are in. And there's different sort of slices of that focus on different types of exposure. So environmental toxicology broadly focuses on exposures in the environment. But more specifically, we can look at occupational toxicology, which focuses on workplace exposures. We can focus on toxic effects of pharmaceutical compounds, and so on and so forth. One area that's unique to the sort of informatics intersection with toxicology is predictive toxicology. And that's when we use computational and statistic techniques to predict previously unobserved toxic effects of specific types of environmental exposures on the human body. And sometimes I need to justify why we care about environmental health science. Again, this might be preaching to the choir, but I like to point out the fact that traditional toxicology that isn't focused on environmental substances, sort of the body of what is mainly out there and where most of the funding is being funneled, is focusing on things like drugs, including approved drugs, but also investigational drugs and the like. Currently, Drug Bank categorizes about 12,000 of those compounds, things that we would consider pharmaceutical drugs. Meanwhile, the US EPA currently catalogs about 2 orders of magnitude greater number of chemicals of potential toxicological concern. And the vast majority of those are completely understudied and have really no data that's available for secondary analysis. And note that this doesn't include any social determinants of health or non-chemical exposures either. So the actual problem space is bigger than that. And we need to start thinking about ways that we can prioritize these chemicals and these exposure types using things like computational approaches to give us a leg up and help us in the basic science setting focus on the things that are most likely to return promising and useful results. So I've said that AI is sort of this area that we work in, and I think that it has a lot of potential to help us prioritize different types of exposures and the effects that they may have. AI is something that's been tossed around a lot lately. I don't think I need to make anyone aware of that. And so just to get us all on the same page with AI can be broken down into many different subdisciplines. So for example, some of those include robotics or natural language processing. There's older techniques like expert systems and automated planning and organization and optimization. Things like computer vision and computer speech are all aspects of AI. The one that I focus on primarily is the machine learning domain. So people often refer to these as synonymous terms, but machine learning is really a subdiscipline of AI that kind of lives and coexists with these other things that are included in it as well. And some of the work we're going to be talking about today includes deep learning, which is yet another subdiscipline of machine learning. So there's sort of this nested Venn diagram of different terminologies. Basically, machine learning is a branch of AI that focuses on using algorithms and existing data to make interesting predictions on novel data that it hasn't seen before. Now, in the last couple of years, one of the places where we have seen AI come up repeatedly is in this notion of generative AI and large language models. So if you've used ChatGPT or Claude or Gemini or these other chat bots in some form or another, any of the myriad applications that have really exploded onto the scene in the last couple of years, these are all fall under this definition of what we call generative AI. And we're not going to get into the technical mathematical definition of what it means, but basically these are a type of machine learning model that can generate novel data due to the mathematical formulation and the training process that we base them on. ChatGPT in particular is the most famous of them, I'd say. It's based on this machine learning AI model called the generative pre-trained transformer. So that's what GPT means. And it's arguably the most successful product launch in history. It's something that all of us are seeing it and its sort of successors impact all of our areas of research. And it spawned an entire industry that makes generative AI sort of this thing, this massive powerhouse that we're seeing. I'm not really going to be talking much about generative AI applications today. We're going to be talking about sort of more traditional machine learning, but that's not to downplay the importance of generative AI. I think it can come into play in a lot of the studies that I'm talking about in various ways, and I'd like to talk to folks about that if they're interested in it. There's lots of these tools that have come onto the scene. This is just to illustrate, even as of early 2023, the field has been very crowded with lots of tools and resources for applying AI. So focusing in a little bit more on the toxicology space now, what is the role that artificial intelligence has in the toxicology space? Well, if you ask me, and different people may have different ideas about this, I see AI as being something that should help experimental toxicologists by doing 2 things. The first is predicting new associations between chemicals and specific endpoints of toxicity. And the second is explaining the mechanisms that might underlie those predictions. So not just saying, we think that this chemical will cause this effect, but we're saying, we think that this chemical will cause this effect in the human body because of these types of molecular and systems level interactions. Importantly, I don't think that AI is currently occupying a space where it should replace experimental validation. Rather, it's going to help us prioritize our time and our effort and our resources to make it so we have a pretty good hypothesis going into any particular research question we want to address. In other words, data-driven science to generate testable, meaningful hypotheses. Okay, so that's sort of what AI is. And now I'm going to hone in on a slightly more specific area of biomedical informatics that's been around since roughly the 1970s, maybe even a little bit earlier, but is becoming, again, much more important as we apply AI to biomedical concepts, and that is knowledge representation. So in order to talk about knowledge representation, we first need to consider this sort of definitional difference between data, which we're all very familiar with, and knowledge. So data refers to raw observations about experiments or the world around us. They can be just observing things outside of an experiment. Often they can be viewed in a quantitative manner. Even if it's qualitative research you're doing, you can consider your data in some sense quantitative. And an example of data would be you do some experiment and you observe specific gene expression measurements after you apply a chemical to human cells and culture. So in other words, you get a specific number saying in this population, we see that this gene is turned on at this level. Knowledge, on the other hand, is a meaningful understanding of phenomena in the world around you. And knowledge is something that results from repeated analysis of many points of data. And it's something that usually happens via some sort of scientific or expert consensus after analyzing it. Typically, knowledge leverages an idealized view of the world. So an example of knowledge that is analogous to my data example would be this chemical X tends to upregulate, in general, expression of gene Y in human cells. That's something that we would determine after analyzing many pieces of data. I'm actually going to skip this slide just in the interest of time. But now that we're talking about data and knowledge, we need to talk about knowledge representations. So knowledge representations are formalized schemas that we can use to represent unambiguously what human knowledge means in a way that sometimes that computers can understand in an automated way, but certainly can unambiguously relay information about human knowledge to other people. So here's a very low resolution image, because it's very old, of a medical knowledge representation from a substance called from something called the Medical Entities Dictionary. Here, what it's doing is it's describing the meaning of plasma glucose as a clinical concept. And it does that by linking it to other meaningful concepts along with these links, such as has specimen, part of, substance measured, and so on, that connect it to other entities within this concept hierarchy. So that's defining the meaning of plasma glucose in a way that a computer can also process. In recent years, we've seen a shift towards this more rigorous way to represent knowledge in biomedicine in particular that uses something that we call biomedical ontologies. Ontologies are something that I'm sure that many of you have encountered in one form or another. For those of you who do research on genetics or molecular phenomena, you may have encountered the gene ontology. That's a way to unambiguously represent things like biological processes and cellular components and the like. And what an ontology does is it defines a hierarchy of biomedical concepts, and then it allows you to rigorously define those in an unambiguous way using relationships that exist between the concepts that are in that hierarchy, as well as additional properties that you can attach to the entities within that thing. If that sounds a little bit abstract, I'll be able to show you a little bit more specifically what this looks like. The way that we operationalize ontologies is sometimes using things that we call property graphs or knowledge graphs. And this is a very, very hot area of AI research nowadays, is the development and application of knowledge graphs to answer specific questions in biomedicine. In this example, we have this rather simple environmental health knowledge graph that just shows the different components of one of these things. We have 3 nodes in this network. Each node in this case represents, there's one chemical, perfluorooctanoic acid. So that's one of these forever chemicals that we find in the water supply, and lots of interest in the media of how these might be affecting human health. We also have at the bottom, we have this gene node representing PPAR alpha, which is a gene in the human genome, as well as a disease node representing the concept of liver cancer over there on the right-hand side of this. We also see that there are these edges which have semantically meaningful definitions to them. So for example, there's an edge connecting the PFOA to liver cancer, and that edge says that this chemical, PFOA, causes the disease liver cancer. That's based on some knowledge we have by observing many points of data. Similarly, we have an edge connecting PFOA to PPAR alpha, saying that PFOA increases the expression of PPAR alpha, and so on and so forth. We also see that we have node properties, which are additional pieces of data or knowledge that we can attach to the nodes or the edges within this property graph that give us additional information that we can use to train and apply AI algorithms to these things. So these are really, really compact, rich representations of complex phenomena that we see in the world around us. And these are particularly useful for predictive toxicology purposes. And that's something that we've been focusing in my lab quite a bit recently. So when we talk about knowledge representation in toxicology, one of the things that we need to explore initially, especially if we're focusing on toxicants, is how do we represent chemical structure data in an unambiguous way? So structures need to be converted to something that's regular and it's computer readable. In this case, I show different ways of representing this chemical called 1-nitro-3-pentafluoroethylbenzene. So we can represent it using its common name or synonyms for the name. We can also represent it using its chemical formula, which just gives us the number of different types of atoms in this chemical that we might find in the environment. Then we have some computationally interesting ways using things called the SMILE string or the INCHI key, or this more regular AI-ready version called a fingerprint. And what the fingerprint does is it asks a bunch of yes or no questions about this chemical structure. So for example, that first question might be, does this chemical compound contain an aromatic ring or not? If yes, then that first bit in this string, this fingerprint string, is going to be a 1, and if no, it's going to be a 0. And so you ask that same set of questions. In this case, it's about 166 of them, I think, for this max fingerprint schema. And it gives you this string of 0s and 1s that you can feed into an AI algorithm. And you can do that for every chemical that's in an analysis, and that's useful for training machine learning purposes. I'll talk a little bit about some of the paradigms for how we do predictive toxicology quickly. The most common and probably one of the oldest of these is something called QSAR, or Quantitative Structure Activity Relationship Modeling. And what this is basically you just build a predictive model. It can be any model that you want. So you can choose a logistic regression model. You can choose a more advanced deep learning model or a linear regression model. And you train it. You train this model and you fit the model to either molecular fingerprints, like I was showing on the previous screen, or other types of molecular descriptors, things like what is the molecular weight, what is the log p-value, what's the number of rings, and so on and so forth. And you train it on data where you know whether it's toxic or not in some sort of in terms of some toxic endpoint. So that when it sees a new unknown material in the future, it's trained to predict with high accuracy whether or not that chemical will also be toxic in the same way. A couple of other analysis paradigms in predictive toxicology are things like read across. This particular slide was provided by Trevor Penning, so thank you for that, Trevor. Basically, the way read across works is that you are trying to impute the toxic value of some target material or the toxic activity. And in order to do that, you look at a number of different read across analogs, they would be called. So these are things that are somehow chemically similar. You can quantify how similar they are, and you look at their known toxicity endpoints. And by reading across this table, you can impute potential therapeutic activities of the chemical that's in question. There's also a sort of more continuous version of that where you do called trend analysis. So this is basically just a regression version. where in this case, the predictor variable shown here on this screen is the N-octanol water partition coefficient. And you see as that particular value for chemicals increases or decreases, does it change the, in this case, LC50 value of that toxic chemical? So does it make it more toxic or not, or less? And then we can go to more rule-based methods like Kramer decision trees, where we have a preset sequence of questions we can ask about any individual chemical that we then step down this tree. So we start at the top and we ask a question like, is the substance a normal constituent of the body, or is it an optical isomer as such? If yes, then we can immediately say that this is likely not a toxic compound, it's low risk. If yes, or if yes, we can say that it's low risk. If we say no, it's not a normal constituent of the body, we need to step down the tree towards question number 2. And as you filter down and ask this sort of 20 questions type sequence, you end up filtering into a specific risk category based on human knowledge about chemicals. We've also seen in the predictive toxicology and the AI for toxicology space, this notion of adverse outcome pathways come out quite a bit. And that is a way of representing adverse outcomes to molecular exposures of toxicants that talks about the different multi-system interactions, which are things we call key events, that give rise to a human or an organismal level or even population level alteration, which is the endpoint of toxicity in this case. So these adverse outcome pathways can form highly complex networks like the one you see on the screen, which are useful in the context of AI algorithms that work with graph formatted data or network formatted data, like I was showing you earlier in the slides. All right, so let's talk about, now that we have all that foundational stuff out of the way, let's jump into some of the research we've been doing in this and similar areas. So one of the big things that we've been working on in my lab is the development and application of this set of resources called ComptoxAI. So ComptoxAI is a large knowledge graph, so a large knowledge representation describing chemicals, xenobiotic chemicals primarily, of environmental toxic concern, different conditions in the human body, so potential endpoints of toxicity, as well as all of the different things that we know in a multimodal fashion that might mediate the relationships between a molecular exposure and the outcome of toxicity. And we build this using data that we've sourced from about 24 different publicly available biomedical databases. And we use one of those biomedical ontologies that I've constructed to restructure and do quality control and normalization of the data that's contained in each of those source databases to construct this knowledge graph and present it in a way that we can then put on a website with a data browser, different access tools for programmatically interacting with the data. So you can query it from your favorite programming language if you do any computational research, as well as some pre-trained machine learning models that are ready for us to ask specific questions that we've already trained on the data and knowledge that's in ComptoxAI. ComptoxAI is available on this site. Some of the features are down as PMAX gets us set up with a new infrastructure as we change our technology platform a little to be something more scalable. But you can access the knowledge graph right now by going to this website and browsing through it. So I encourage you to do so when you have some time. So what are some of the things that we can do with this? A few of you, if you've heard me talk before, might have seen these examples already. But these are some nice illustrative examples of what you can do with a knowledge graph. So one of the first things you could say is you say, well, what are the shortest set of paths in this graph that link two different things together? In this case, let's look at that example we were looking at earlier, which is PFOA, perfluorooctanoic acid, that forever chemical that's found in the water supply, and non-alcoholic fatty liver disease as one of the things that we know is increased in risk when you're exposed to high levels of PFOA. This is one of the sort of newer indications resulting from research on forever chemicals. So we're still trying to figure out a lot of the mechanisms that underlie this risk association. So what we see ComptoxAI tell us is that exposure to PFOA results in either decreased expression or increased expression of a number of different genes that are found in the human body, including that PPAR alpha, which I was showing in the mock-up earlier, and a bunch of other things, all of which are then associated with increased risk for disease. So through a bunch of different avenues, it's exerting different gene regulation effects that likely are giving rise to the increased risk for non-alcoholic fatty liver disease. Importantly, several of these, specifically this NFE2L2 and CD14 genes, have never been co-described with both PFOA and non-alcoholic fatty liver disease in the scientific literature prior to us showing this in this study. So what that means is that by aggregating information from multiple databases, we're able to make and imply potential mechanisms that lie between a chemical exposure and a toxic endpoint. We can also explore the structure of neighborhoods that surround a given node of interest. In this case, we're looking at the chemical dioxin, which is familiar to toxicologists as being a highly toxic chemical that leads to some severe health effects, including death, and has been associated with a number of industrial incidents over time. We can see that dioxin is sort of associated with these clusters or groups of adverse outcome pathway type entities and concepts that tend to be sort of talking about here in this case in group A. We are seeing things sort of mainly related to reproductive health outcomes. C and D seem to be sort of cancer related, and B looks like it might be kidney related. And we see that there's these interesting connections that link those different regions within this network together that might be exploitable for targeting therapies. Also, we notice that there are other chemicals that tend to creep their way in. So all of the other red nodes in that network represent different chemicals aside from dioxin that for the most part are extremely understudied, or we don't know as much about them. So we may be able to use what we know about dioxin, which is very well studied, to imply potential toxic and therapeutic indications about these other chemicals that we have not yet observed. So getting into the more AI applications of this, we wanted to see, can we use this ComptoxAI knowledge graph to improve upon this very old technique that is QSAR? QSAR doesn't tend to perform very well. It's only able to make the correct prediction about toxicity like 60 to 70% of the time. So we wanted to see if ComptoxAI could do better. So the way we did this is we extracted what's called a heterogeneous graph consisting of chemicals, genes, and assays from the graph database in ComptoxAI. And we trained a particular model that's called a graph neural network, or a convolutional graph neural network. We trained this thing based on known associations for 52 different endpoints of toxicity, and saw whether or not it was able to accurately make predictions on a separate held out set of chemicals that we didn't train these models on. And this, like I said, is using something called a neural network model, which is something that's associated with deep learning. These are basically predictive models that approximate biological neural networks. They consist of these different nodes, which we call neurons, organized into different layers, and they have different connectivity patterns between them. So we see different inputs flow into certain nodes in the network, and based on the strength of those inputs, it will either fire or not fire and propagate that signal to other nodes in the network. And by arranging these in sufficiently large and complex orientations, and by training it so the parameters associated with each of these nodes have different intelligently populated values, these neural networks can make very, very complex decision-making procedures. So the type of neural network that we focused on is one called a graph neural network. And specifically, that's a neural network that's designed to work with graph formatted data like what we have in ComptoxAI. So let's say we want to make a prediction about this node here, which I'm calling a disease node. And this is just a mock-up. It's an illustrative mock-up. It's not an actual graph. But in order to make a prediction about that disease, it would first look at its immediate neighbors. The second layer of the neural network would then look at their neighbors, and so on and so forth. So in other words, it's making predictions about this disease based not only on the traditional data that we usually have available for making predictions, but also this complex network data and the topology of that network to impute new activities. So the question is, how did this new model that we made for predicting the toxicity of certain chemicals work in comparison to sort of the existing models? And it turns out that in every case we tested, it worked remarkably well. So these are just two of the toxic endpoints we tested. We have pregnane X receptor agonism on the left side and HepG2 cell viability, so in other words, cell death on the right-hand side. And this is what we call an AUROC plot. Basically, the area underneath the curve is proportional to the performance of the algorithm. So the red curves, which are does somebody have a question? All right. By the way, feel free to interrupt if anybody has anything. I'm totally happy to stop if any questions do come up along the way. Basically, we see that the red curves in these graphs, and this is true in all 52 of the ones we did, not just the ones I'm showing here, had significantly greater area under the curve than the sort of traditional gold standard models, which were random forest and gradient boosting classifier models. And what that means is that in every instance, the graph neural network did a better job at predicting toxicity. So we did something that's common to AI studies called an ablation analysis to figure out why it's performing so much better. And it turns out that if you remove from these networks either the other genes that are in there, so this gene information, or if you remove the structure, the chemical structure, which is usually all that QSAR has to go on, we just barely see the performance drop a little bit. However, if we remove the other toxic endpoints that are visible to the model, so in other words, unrelated endpoints of toxicity where we know their value, we see the performance drop off to just barely better than randomly guessing yes or no. So in other words, what these networks are doing is they're using known implications, existing knowledge about toxicity to impute other types of toxicity with highly accurate and highly precise results. So moving a little bit into some other things that we've done with this. Like I said, we want to make our predictions in toxicity interpretable and meaningful. So one of my students, an undergraduate student who did an internship with us a couple of years ago, Estefania Morales, she from Tufts University, she trained what we call explainable AI models that look at the different chemical characteristics within a predictive model, and they tell us what the most informative aspects of chemical structure are that tend to be focused on by the machine learning for making a specific prediction. So for example, we're seeing that a lot of characteristics related to aromatic rings are showing up in these machine learning experiments for, in this case, food contact chemicals, activating a liver-based assay, or this list of general hazardous substances provided by the EPA and colon cancer sensitivity. We see that they tend to be focusing on the presence of aromatic rings, and that was a very common thing that we saw. And we've released the code for doing this, and it's freely available on GitHub if people want to apply it to their own chemical data sets. Also, from a more advanced AI standpoint, we can start actually piping the actual structure of specific molecules into the ComptoxAI model. And this is work done by a PhD candidate, or actually he just received his PhD. He just defended successfully. His name is Giuseppe Albi at the University of Pavia, who visited our lab to show that if you take the knowledge that's in ComptoxAI and then you add a formal representation of chemical structure, we can perform even better on this QSAR approach, and we can start to get explainable outputs telling you which regions of the chemical the AI algorithm is focusing on when it's making a prediction that this is toxic or not. So it can start to talk about the active regions of chemicals that are likely binding to something at a molecular level and conveying that activity. Now, I want to talk for the rest of the time about some applications in environmental maternal health in particular. What we've been talking about so far is much more methodological and foundational that can apply to kind of any area of toxicity. But we're particularly interested in adverse outcomes during pregnancy. This is sort of a schematic that outlines some of the types of experiments that we're beginning to ask in our lab. In this particular proposal that is being considered by the NIH right now, we are taking the information that's contained in ComptoxAI, so this structured biomedical knowledge, and integrating it with the data that we have in our EHR database here at Penn, along with other public health data streams, ancillary clinical data sets, and other interventional data sets to result in what we refer to as an observational exposome knowledge graph. So this is what we are able to see of the human exposome in a longitudinal fashion. So all of the exposures that are tracked in the EHR as well as in public health records for an individual person that represents their kind of environmental health over time. Now we can take these knowledge graphs representing a person's exposome and we can feed them into specific machine learning models, which we call temporal graph machine learning models, to do things like predict either preeclampsia or gestational diabetes in this case, and focus in using explainable AI, both for looking at lifetime risk factors for disease, as well as using something called temporal graph attention, which is using a similar mechanism to how large language models work, to also identify windows of susceptibility for those specific outcomes of pregnancy. So in other words, certain risk factors which might map to specific chemicals or social determinants or other types of risk factors we identify may have either harmful or protective effects in terms of that maternal health outcome during different time points and time windows aligned along the point of and immediately preceding a pregnancy. So we're working on building the framework that will support this larger effort. A recent paper that we did publish that sort of serves as great preliminary evidence for this work comes from this work that one of my PhD students, who's co-advised by Shefali Sethi-Verma, published at the Pacific Symposium for Biocomputing in a work that's titled Drive KG. And in this, she used variant phenotype association discovery in complex diseases using heterogeneous knowledge graphs. So the motivation for this is that complex diseases, such as diseases associated with maternal health, are often governed by both genetic variation and external influences. And so the biological context of genetic variance is very important, and we often don't consider that when we're making predictions using things like polygenic risk scores or other genetic sources of information. So heterogeneous graph neural networks and knowledge graphs, like the one that we have in ComptoxAI, provide a very robust mechanism to integrate these diverse factors together and consider them simultaneously. So we want to see that we have greater predictive power, improved explainability or interpretability, or both of these things. And so we looked particularly at endometriosis, which is not maternal health, but it is a highly important women's health condition. It's a highly complex disease that stands to benefit from this, and it's highly understudied. And we also wanted to see if we can help elucidate shared mechanisms with other diseases that may be highly comorbid with endometriosis. And the way that we did this is we defined a knowledge graph, basically, which is a template for individual patients that we would represent in this study. We'd populate that knowledge graph using entities from 8 different openly available databases that are freely available to anyone in the public. And then we trained a heterogeneous graph neural network to predict missing links between specific genetic variants and phenotypes, which represent disease. So like I said, we selected endometriosis and then its comorbidity obesity for these 2 phenotypes, using a specific approach using things called node-to-vec and memgraph as a framework. And then we generated individual patient graphs using highly associated SNPs that we identified using that graph neural network, and trained a different type of machine learning model to classify each patient graph based on whether they were either sick or healthy in terms of, in this case, endometriosis or obesity. So this is what that sort of schema looks like. It tells you the different types of entities that are considered in the knowledge graph. We had genes, we had SNPs, which are variants. We had phenotype, which was disease, and then we had proteins, which is the resulting transcript of the gene, which can be associated with what are called PQTLs. And then these are the different databases that we retrieved the publicly available information for building these knowledge graphs from. Both of the nodes, so the actual entities, as well as the edges that link those nodes together. This is all reproducible. We have all the code posted online so other people can build these if they want. And so we were able to start looking at things using this, this heterogeneous knowledge graph to start identifying highly relevant associations with, in this case, endometriosis, and then also with obesity, showing that there are different genetic markers that are highly associated with it. Many of them are new signals, meaning that they were not identified in existing GWAS studies, showing that we have the ability to identify with greater predictive power different associations based on this knowledge graph approach. And we did various sensitivity analyses, and we show that the distributions of these predictions hold up across different disease contexts, and so on and so forth. So the takeaway points from this study are that heterogeneous knowledge graphs and graph machine learning identified new SNPs, new single nucleotide polymorphisms associated with endometriosis. These new single nucleotide polymorphisms tend to be enriched for variants that are linked to a number of different things, including body mass index, lipid metabolism, and depressive disorders. All of these are highly comorbid with endometriosis, as is obesity. And by visualizing these networks, we showed connections to underlying inflammatory and cell proliferation pathways, which suggest shared disease mechanisms between those diseases. So these results help explain something that's known as the missing heritability problem between traditional polygenic risk scores and individual heritability estimates from traditional twin studies. So we do see an increased boost in the classification risk as well, sort of as an added bonus. We're able to make these predictions slightly better and more accurately. That's a moderate boost, but it is meaningful. So the most important takeaway is that we were able to identify new associations that can explain comorbidities as well as the mechanisms underlying disease using this knowledge graph approach. So shifting entirely to a different type of analysis, focusing more on public health data and area level measures of environmental health, I want to highlight work done by my PhD student who's actually on this call. Thank you for joining, Chloe. Chloe is a PhD candidate. I know that Ayman is also very familiar as one of her committee members. In Chloe's work, she's been looking at different exposures that occur at the area or neighborhood level, in this case, in the city of Philadelphia, and seeing how those associate with preeclampsia. And what we were able to identify is that there are specific census tracts within the Philadelphia boundaries that are highly associated with preeclampsia as a maternal health condition, some of which seem a little bit paradoxical and pose some interesting questions that we want to explore in the future, but showing that there are patterns that we can leverage either for policymaking or prediction or other types of applications between things like social determinants of health, area level environmental exposures, and other things that impact the human exposome. And I do want to mention also that Chloe is expanding this. I did not include the results here because she's still working on finalizing them. For looking at the entire state of Pennsylvania and associations between different maternal health outcomes and environmental exposures at the statewide level to see how this behaves, not just in Philadelphia, but across the entire Commonwealth of Pennsylvania. And I want to thank Ayman and his research group for helping us with some of the data access for that project. So we'll be able to show that off soon once Chloe has the results a little bit more finalized. Okay, so that's just a quick tour of some of the questions that we're starting to ask, and it might give you some ideas of things that we can do with AI related to maternal health, as well as sort of more generally predictive toxicology and environmental health science. And I want to leave you with just a couple thoughts for the future. Again, this is where I get up on my soapbox for a couple of minutes. The first here is that I see that we are shifting to an era where we can start to define research being knowledge-driven AI. So this is something that I made up myself. And since I started talking about this at talks, I've seen a couple blogs refer to it. So I hope that those are people who heard me define this. But basically, this is an extension of what many of us know about in data-driven science. Data-driven science is where we say that you don't want to go into an experiment with a possibly biased, preconceived hypothesis about what you want the outcome to be. Instead, you should apply your analysis to a large body of data or a large body of indications or associations and let the data tell you what you should focus on. So this is, in a sense, a way to generate new hypotheses that can be subsequently evaluated, and those are not based on more biased approaches to generating hypotheses. And this is also increasingly accepted by a broad range of domains as a valid way to do science, not just the traditional hypothesis-driven approach. For example, NIH reviewers are less averse to this type of way that we do research, and they're seeing the value. Knowledge-driven science is an extension to data-driven science that incorporates structured semantic knowledge like the kind that we have in ComptoxAI. And this uses what we already know to focus and refine the results of what we can tell with data. An important side effect of this is that knowledge-driven science facilitates explainability of the results we see. So if we can map the results back to these human-readable, understandable explanations of things, like in the knowledge graph, we can naturally understand what is likely underlying those predictions. And then knowledge-driven AI, or knowledge-driven artificial intelligence, is when you use AI techniques to accomplish this knowledge-driven science. So we've been developing a sequence of different software tools and resources for the scientific community that will enable them to construct their own knowledge representations in their own domains of research. And these are things that are publicly available. They're on GitHub and they're under active development. And so I encourage you to take a look at some of the stuff that we are doing. This is something called ISTA. So this is an ontology guided toolkit for knowledge graph assembly. And for those of you Lord of the Rings nerds, ISTA is based off of the Elvish word for knowledge. So just giving a shout out to Tolkien there. There's also a lot of different resources online showing you existing knowledge representations for all different kinds of domains and applications. So I encourage you to go out and look at specific domains that you're focusing on and seeing if somebody's already made a publicly available knowledge graph that you can then apply knowledge-driven AI to your own research. The last thing I want people to think about is, are we on the verge of another AI winter? This is something that comes up repeatedly when we talk about artificial intelligence in 2026. Do we think that AI is here to stay and it's something that is going to be around, or do we think that it's going to go to the wayside? Well, Bloomberg's making predictions saying that there's another AI winter on our horizon, meaning that people are going to lose interest and not be using AI as much. But I think that it's here to stay in some aspects. It's found its way into so many different aspects of life and research that I think that we are going to be coping with how we responsibly and ethically use artificial intelligence in our research. And also in biomedicine and informatics, we've been using AI ever since the 1960s when it was first defined. So my prediction is that while in the public sector we may see some waning interest, it's around and it's here to stay, and it's something that all of us are going to have to figure out how can we use responsibly and in ways that encourage good science to be done. And last but not least, I will say that biomedical informatics is sort of one of the ways that we can encourage responsible use of AI. So I want everybody to think about what is the purpose of biomedical AI. I don't think that there's a right or wrong answer, but everybody should be thinking about this. So what are the things that we need to be concerned about over the coming years? Things like funding opportunities and other policies and different ways that we can use certain tools in different types of research. We need to think about how can we do that responsibly and ethically. And I think that keeping AI grounded in existing rigorous knowledge will ensure that it always leans towards making better scientific discoveries. And even though there's this whole financial aspect to AI right now, invariably, I think that it stands to improve our understanding of human health in the natural world if we do it responsibly. So with that, I will wrap up. I want to thank my students who participated in the work that I was able to highlight today. Again, I want to thank the organizers of the talk for inviting me to be here. And then, of course, my collaborators and the different funding institutes as well.